Well, I'm as ready as anybody to be enthusiastic about new things. But first, I want to separate the hype from the reality. Well, hello and welcome to another episode of You and Me and Multiple Sclerosis. My name is Pam. I've been living with multiple sclerosis now for 39 years. And in that time, I've seen new ideas come, and some of them stick, but most of them go. And nowadays, when I see a new idea, a new treatment, a new medicine, a new option, I'm a little bit wary. I always want to check into things and make sure that it's worth getting excited about before I get excited. And you've seen that happen here a lot on this channel. And I realize now that probably what I could be doing is helping you see my process a little bit, because part of what I like to do is help you figure out how to make some of these decisions and discoveries for yourself. And it just so happens that there was an article that just came out the other day about a new DMT, a new disease-modifying therapy called Frexalimab, and I don't know if you've heard anything about it yet. It's only completed its phase 2 trial, but there are quite a few people that are saying that it's the greatest thing since sliced bread, etc. And of course, that always gets my antenna up because I want to know more about this and see if it really is everything it's cracked up to be. So why don't we go ahead? We'll take a look at the article first. And as I go through it, I will show you, as I'm reading an article, what kinds of questions might I have. And then from there, we go through a process of discovery to see how much of the claims of the article seem to hold water or to be universally held by other experts in the field that I respect. Another thing that I like to do is just see if there's a clinical trial that's been posted. I might be able to go out and look at that and gain some information from that. Also, any other related clinical trials or follow-ons from the one that's been published. What can I learn from that? And then, as I say, I go out and I look for other authorities who have weighed in on this new DMT or this new idea to see what they're thinking. Am I getting any of my questions answered along the way? And then finally, I pull all that together and I arrive at least a tentative conclusion, sort of a working conclusion, if you will, because obviously there'll be more information coming down the pike eventually. And I've got to be ready to change my opinion one way or the other if the information passes the sniff test, as it were. But this will help you understand what my process is. Why don't we go through this new article that's just come out about Frexalimab, and I'll step you through some of the other pieces that I looked at so that I could figure out what I should think about this. This is the article that I saw out of MedPage today on February 14th of 2024, so just the other day. It's called Novel Anti-CD40L Antibody Slows New Brain Lesions in Relapsing MS. And the drug is called Frexalimab. It's a monoclonal antibody, so it would be similar to Ocrevus, because the technical name of Ocrevus is ocrelizumab. And then I'm sure that there are a number of other monoclonal antibodies that are being used right now for MS therapy. But they say that this new one shows promise in phase 2 studies. It goes on to say that Frexalimab, an investigational second-generation CD40 ligand inhibitor, led to fewer new gadolinium-enhancing T1-weighted lesions in relapsing multiple sclerosis, a phase 2 trial showed. Okay, well, I have two questions right off the bat. It does say that there are fewer, so that means there are some lesions. And the other is fewer than what? So I guess hopefully the article will address that. I'm not sure that we're supposed to be excited yet about something that doesn't actually stop the lesions altogether, because we do have a number of DMTs that slow lesions down considerably. So why is this one superior? I guess we'll hopefully find out. At week 12, they say, the adjusted mean number of new gadolinium-enhancing T1-weighted lesions was 0.2. In MS patients who received 1,200 milligrams of Frexalimab intravenously and 0.3 in those who received 300 milligrams of Frexalimab subcutaneously, compared with 1.4 in those who received placebo, reported Patrick Vermersch, MD, PhD, of the University of Lille in France, and co-authors. And I will be pulling up the paper in a while by Patrick Vermersch and co-authors. Compared with placebo, rate ratios were 0.11 for the 1,200 milligram group and 0.21 for the 300 milligram group, the researchers wrote in New England Journal of Medicine. So one thing I do want to point out is New England Journal of Medicine is a highly prestigious journal, and I'm sure that the peer reviewers would not publish something unless it passed muster. So that's one way to help us feel good about the research that's being published. Secondary imaging endpoints generally were in the same direction as the primary endpoint, Vermersch and colleagues noted. So in other words, they had a primary target that they wanted to hit, and they also had some secondary targets, and they looked like all of them were hitting about where they wanted. Plasma levels of neurofilament light, a biomarker of axonal damage, appeared to decrease in the Frexalimab groups. Appeared does not mean it actually did, it just means the data that they collected supported that it decreased. The most common adverse events that occurred in the trial were SARS-CoV-2 infection and headaches. Well, nothing life-threatening, but nothing to sneeze at either. And we don't know why those were. We don't know that the drug caused it, or if the infections were going to happen anyway, the headaches would have happened anyway. We don't know that. At least not from where we're sitting right now. The scientists may have more data about that. Given that this is just a summary of the research, we don't know, at least not yet. The paramount need in MS is for a more effective therapy against disease progression, noted Stephen Hauser, MD of the University of California of San Francisco, in an accompanying editorial. Okay, well, that's very good to know. If Stephen Hauser is behind it, he also is somebody that I have a lot of respect for, so that makes me feel better. Even though I'm still, like I say, the jury's kind of out yet. I'm wondering why do we need this one? Because I'm not even sure that I would agree that the paramount need in MS is for more effective therapy against disease progression. Yes, that's an important need, but the paramount need is to stop it from happening at all. And we aren't there yet. Okay, they go on to say, highly effective drugs in relapsing MS, like the anti-CD20 agents, have shown that when relapses are prevented, progression independent of relapse activity is uncovered, Hauser observed. Thus, progression is present in most, if not all, patients with multiple sclerosis, regardless of where they are in the disease continuum, he wrote. And yes, and that's exactly why I said earlier, the paramount need is for something that will stop MS from happening at all. They go on to say Frexalimab is an anti-CD20 ligand antibody that blocks the co-stimulatory CD40 ligand pathway. Not that we are expected to understand that. Because of its role as a key co-stimulatory molecule that mediates interactions between T lymphocytes and B cells, as well as other antigen-presenting cells, blockade of CD40-CD40L engagement is a rational therapeutic strategy in multiple sclerosis, Hauser suggested. Inhibitors of the CD40 pathway could downregulate innate immunity in the central nervous system. And remember that innate immunity is the immunity that we are all born with. A major contributor to progressive multiple sclerosis, he pointed out. Although the current trial was neither designed nor powered to assess benefits against progressive multiple sclerosis, progression is where the true clinical value of Frexalimab and its place in the therapeutic armamentarium against multiple sclerosis will need to be defined. Absolutely, I would agree with that. The phase 2 trial included 129 people with relapsing MS, 125 completed the study's 12-week double-blind period. So this was indeed a double-blind trial, and it looks like there was probably a placebo in here too, so it would have met the gold standard of how you do a clinical trial these days. It says that the mean age of participants was approximately 37 and 66% were women. About a third, or 30%, had gadolinium-enhancing lesions at baseline. All participants had at least one relapse within the previous year, or at least 2 relapses in the previous 2 years, or at least one active gadolinium-enhancing lesion in the 6 months before screening. Patients with secondary progressive MS who were having relapses could enroll. Overall, 94% of participants had relapsing remitting MS and 6% had secondary progressive disease. The median expanded disability status scale, or EDSS score, which ranges from 0, normal exam, to 10, death due to MS, was 2.5, so very low. These folks were early in their course of disease, apparently. And I just will point out that the reason there's mostly relapsing MS here and not progressive MS is because the trial had to prove its effectiveness, and in order to do that, there had to be relapses going on so we could see whether the drug would slow that down. Participants were randomized to receive 1,200 milligrams of Frexalimab administered intravenously every 4 weeks after an 1,800 milligram loading dose. 300 milligrams of Frexalimab administered subcutaneously every 2 weeks after a 600 milligram loading dose, or matching placebos for each treatment. After 12 weeks, all the participants could receive open-label Frexalimab. Well, that's pretty neat. The primary endpoint was the number of new gadolinium-enhancing T1-weighted lesions on MRI at week 12 relative to week 8. Secondary endpoints included the number of new or enlarging T2-weighted lesions at week 12 relative to week 8, the total number of gadolinium-enhancing T1-weighted lesions at week 12, and safety, of course. The trial was too brief and small for conclusions regarding clinical outcomes, but over the 12-week double-blind period, no relapses occurred in the group that received 1,200 milligrams of Frexalimab intravenously, and relapses occurred in approximately 4% of the participants in the group that received 300 milligrams of Frexalimab subcutaneously and in the pooled placebo group. Vermersch and co-authors noted. Study limitations included the trial's small sample size, short duration, and use of imaging endpoints. Preliminary data from the open-label extension period suggested Frexalimab continued to have a sustained reduction of MRI activity in 6 months. Phase 3 studies in relapsing MS and non-relapsing secondary progressive MS are ongoing. So there you have it. So that's where things stand. It's only a phase 2 trial, and there's going to be at least one more phase that's going to look at a wider range of participants with various courses of MS. And hopefully they're going to be able to get some data from longer term than just the 12 weeks of the study. And this is the article itself, which I will not go through with you here, but I'm linking it down below so that you can, if you wish to, because this is part of my process. I look through the actual paper, especially I look at the abstract and the conclusions just to see if it matches up to the article that I read. Instead, I'm inserting a short video from the New England Journal of Medicine group about the paper itself.
Patients with multiple sclerosis, or MS, have high levels of soluble CD40L that broadly correlate with disability. Clinical and pathological observations have suggested targeting CD40L inhibition as a potential therapeutic strategy. A new trial evaluated the efficacy and safety of the second-generation anti-CD40L monoclonal antibody Frexalimab for these patients. The drug was designed to avoid thromboembolic events that had been seen with other drugs in this class. In this Phase 2 double-blind trial, 129 patients with relapsing MS were randomly assigned in a 4:4 to 1:1 ratio to receive 1,200 milligrams of Frexalimab intravenously once monthly with an 1,800 milligram loading dose, 300 milligrams of Frexalimab subcutaneously once every 2 weeks with a 600 milligram loading dose, or matching placebos. The primary endpoint, the number of new gadolinium-enhancing T1-weighted lesions at week 12 relative to week 8, as determined by MRI, favored Frexalimab treatment. With an adjusted mean number of new lesions of 0.2 in the 1,200 milligram intravenous group and 0.3 in the 300 milligram subcutaneous group, as compared with 1.4 in the pooled placebo group. Secondary endpoints, the number of new or enlarging T2-weighted lesions at week 12, and the total number of gadolinium-enhancing T1-weighted lesions at week 12, generally favored Frexalimab. The most common adverse events, in 5% or more of the participants in any group, were COVID-19 and headaches. The authors conclude that in patients with relapsing MS, Frexalimab showed a greater reduction in the number of new gadolinium-enhancing lesions between weeks 8 and 12 than placebo. Full trial results are available at NEJM.org.
So maybe you caught that, but right in the beginning, they talk about thrombolytic events or blood clots reduced with this drug compared to other monoclonal antibodies. So that is a good thing. So going out to the clinicaltrials.gov website, I searched for multiple sclerosis and Frexalimab to see what I could find. And I found several studies actually that are actively recruiting, well, 2 that are anyway. This must be a follow-on. It's on efficacy and safety study of Frexalimab in adults with non-relapsing secondary progressive. And that is a very important study because remember the phase 2 trials that just finished were on people who were in some form of relapsing, either relapsing MS or progression with relapses. And this is a way of looking at the non-relapsing secondary progressive, which I think is really pretty neat. I don't know how they're planning to go about giving the study long enough time to see if there's any good effect, but I'm curious about it. And as you can see over on the far right, they've got places in California and in Florida. And if you're near any of those, well, next looks like there's 9 more. So you can go out and look and see who else is recruiting. So maybe you could participate in the study. There's another one, efficacy and safety studies of Frexalimab in adults with relapsing forms of MS. So that's similar to what they were already working on. It looks like they're using it in conjunction with teriflunomide. That's interesting. Okay. So there's a place in Scottsdale, Arizona that's recruiting, and there's a place in California, place in Florida, and 7 more. So once again, you can go out and get involved in that if you want to. So there are clinical trials going on, not very many. I found 3, but there are clinical trials happening, and I think that's pretty good news. I just wanted to look at the editorial by Dr. Stephen Hauser that was actually referred to in the article we just looked at a little while ago, Silencing Immune Dialogue in Multiple Sclerosis. And the way his comments were cited in that article, it certainly gave the impression he was very excited about this and very positive about it. But, this is a good way of verifying that that's really true. So this was published February 15th of 2024. And I'm not going to read all of it, but I just cut and pasted a few things out of it, and I thought I'd go through those. He says, what can we conclude from these preliminary data from the phase 2 trial? The results appear clear, although the clinical significance is uncertain. Clear because there was an unambiguous benefit with regard to short-term MRI outcomes in patients who received Frexalimab as compared to with those who received placebo, and because a generally low level of MRI activity persisted during an additional 12-week open-label extension period. A similar decrease in MRI activity was also observed in patients with the placebo group who crossed over to receive Frexalimab therapy in the extension period. Positive results with Frexalimab were supported by the lower levels of neurofilament light chains in peripheral blood, a biomarker of brain tissue damage. If these benefits are confirmed with longer-term use of the agent, it seems likely that there will be a protective effect of Frexalimab therapy with regard to relapses of multiple sclerosis. Nevertheless, some questions have not been addressed. Well, I was happy to see that because I actually have some questions. I don't know if he's going to get to them, but let's find out. He says that the striking clinical benefits and safety profile of the available high-efficacy therapies for relapsing multiple sclerosis create a high bar for any new treatment. In this trial, the MRI outcomes with Frexalimab therapy were impressive, but appear to be less complete than those with anti-CD20 agents, although trials of different agents cannot be directly compared. Safety with long-term use is another unknown. Even if future studies show Frexalimab to be competitive with currently available therapies for relapsing multiple sclerosis, the paramount need is for more effective therapy against progression. So he's repeating kind of what we saw in that article, that there's a paramount need for dealing with progressive MS, although he's not as specific as the article, and I would agree with him as far as progressive forms of MS do need more attention, but the article implied that we needed something that was going to slow progression, whereas I think what we really want is something that's going to stop progression, that's going to keep us from ever getting to the stage of MS where we are progressing. And then finally, he says, although the current trial was neither designed nor powered to assess benefits against progressive multiple sclerosis, progression is where the true clinical value of Frexalimab and its place in the therapeutic armamentarium against multiple sclerosis will need to be defined. So there you have it. I don't see this as a negative review at all, but I don't see it as unconditionally positive as I was thinking it was based on how it was referred to in the article. One of the first people to weigh in on this was Professor G, Dr. Gavin Giovannoni. He had a MS Selfie podcast on that, and I'm going to play some excerpts from that so you can see what his take is on Frexalimab and what it adds to our MS armamentarium.
I'm incredibly excited by this publication, and I think it's enormous or very big news, and it actually has rejuvenated my enthusiasm for the future of MS treatments. And the results are incredibly positive. When you look at the high dose, it reduced MRI activity by 90%, which puts it in the very highly effective therapy bracket. And over time, their therapy got even better. And this trial was very short. It was essentially a 3-month trial, simply because it's becoming increasingly difficult to put people into placebo-controlled trials for longer than this period of time. So I think the results are incredibly important. And not only did it have an impact on MRI activity, but it also reduced blood neurofilament levels and an inflammatory marker called CXCL13 quite profoundly in a very short period of time. And it also had an impact on quality of life. So this is remarkable results for such a short study. I also think that going beyond just the results is telling us something really important about the pathogenesis of MS, and it confirms the critical role of T cells in the pathogenesis of MS, because this particular pathway is critical at activating T cells. And so it's suggesting that MS is an autoimmune disease. And the problem I have, and it causes cognitive dissonance, is that I've taken a very strong position on EBV being the cause of MS, and if the EBV is the cause of MS, how does this trial result with an anti-CD40 ligand explain Epstein-Barr virus driving MS disease activity. I'll also take the position, even if I'm wrong about EBV and Frexalimab, the results of this study are sending a real buzz through the MS community, particularly amongst immunologists, and actually also genomics experts, because both the immunologists and the genomics experts predicted that the CD40 ligand is critical in MS pathogenesis and predicted these results 2 decades ago. Now, the reason why the first generation of monoclonal antibodies against CD40 and CD40 ligand failed is because they tended to activate platelets and cause thrombosis. We now know about that, and it's not to do with it's to do with the what we call the FC fragment of immunoglobulin, and by engineering out the platelet binding domain of those antibodies, you actually prevent that from happening. So this new generation of anti-CD40 ligand antibodies, including Frexalimab, have not got thrombotic complications, which is reassuring. So this drug is not going to be, in my opinion, associated with thrombosis. Anyway, I was asked by a colleague yesterday when these results came out, where do I see the MS market in 10 years' time? He doesn't think there's space in the market for another anti-inflammatory like Frexalimab. And I think, his opinion is that because of the stunning results of the anti-CD20, their relative safety, ease of use, that there is no space for another anti-inflammatory. And so I disagree with him, and I suggest that he read my extensive commentaries on anti-CD20 therapies. It's very clear that anti-CD20s are very effective at suppressing relapses and focal MRI activity. In other words, you become inactive with no evident inflammatory disease activity, but it doesn't stop. These therapies do not stop smoldering MS. So a significant proportion of people on anti-CD20s will continue to get worse because it's not targeting CNS mechanisms and what's driving smoldering MS. And also, I don't think people can stay on anti-CD20 therapies lifelong. A proportion develop hypergammaglobulinemia, the immunoglobulin levels drop. And as you age, the risk of infection increases. And we are seeing an increasing number of patients being admitted with severe infections. So we definitely need an exit strategy from anti-CD20 therapies. And Frexalimab may be one of those exit strategies. And so if the phase 3 Frexalimab studies show the drug to have a much more pronounced effect on the end organ, in other words, reducing brain volume loss, people develop fewer paramagnetic rim lesions, fewer slowly expanding lesions. There's a reduction in spinal fluid biomarkers of both B and plasma cell activity, less microglial activation. Then Frexalimab will win hands down. Who wouldn't want to go on to the therapy that reduces or prevents smoldering disease? So I think this issue about is there space will depend on how good Frexalimab proves to be in phase 3 trials. So we shouldn't second guess that. And there's also this very tantalizing possibility that Frexalimab may reestablish immune tolerance, and it won't need to be given continuously. So there is this theory that Frexalimab and this class of drugs may reestablish immune tolerance and switch off autoimmunity. In other words, cure MS. Get rid of the autoreactive T cells. So I think we should wait to see what the results are, and the idea that you have to stay on Frexalimab lifelong may not be necessary, because if it does reestablish tolerance, you may only need a short course of it. It's clear to me, and it should be clear to my colleague and my colleagues in general, that anti-CD20 therapies have a limited shelf life as a treatment for MS in the way we use them now. And I don't think they will be the mainstream of MS treatment in 10 years' time. And you may not be aware, but all the big pharma companies that have licensed anti-CD20 therapies are not sitting on their hands. They all have very deep and very interesting MS development pipelines. And this tells us from within the pharmaceutical companies that have these therapies that they are not good enough. And and it's clear that we have to go beyond just preventing relapses and suppressing focal MRI activity. And, we have to get to the smoldering MS. And so the most exciting of these products at the moment are the CNS penetrant therapies targeting smoldering MS, and this includes CD19 targeted CAR T cells and proteasome inhibitors like Exozumab.
Well, I hope you caught what Professor G was saying there. Not only are there fewer side effects with this, Frexalimab might even cure MS? Really? Wow, that would be truly amazing. Well, I hope that it was helpful for you to see the whole process that I go through. I do this pretty much for everything that I encounter as I'm researching topics for this channel, because I do not want to put anything out on this channel that has not been tested and has not been given some consideration. I'm not going to join in with everybody else either on the hype side or on the gloom and doom side, because everything is a mix, right? Nothing is perfect. Nothing has all the answers, but nothing is without merit either, because sometimes things are just a step in the direction that we want to go, but we're not there yet. And I think that we've seen that many times with many of the new disease-modifying therapies that have been promoted and some of the non-pharmaceutical options that have been promoted. There's good and there's maybe not quite there yet in every single one of them. And I hope that this will help you make some of those decisions for yourself when people tell you about a new thing or when you read an article about a new thing. Maybe this way you can be ready to engage with that idea a little bit, be a little more critical of it. It might even help you when you're meeting with your doctor who is recommending certain medications. You can have some questions to ask that will help you make a better decision. But that's all I have for you today. And so I would love to see you take really good care of yourself. It's so important to do that. We've got to stay healthy as much as we can. Keep our quality of life as good and strong as it possibly can be. So that's your assignment until I see you again in my next video.